right, guys, in this lesson, we're going to look at nerve impulse transmission. So this impulse transmission could be from neuron to neuron, or it could be from neuron to muscle. So with neuron to neuron transmission, we can either have excitatory or inhibitory signals. Excitatory means that it keeps that action potential going. It continues to send the signals or creates an action of some kind. Inhibitory is exactly the opposite. It means that when that signal, that action potential gets there, it actually stops it. So it inhibits sending the signal any further. Now, when we see neuron to muscle, we only see excitatory because it's actually causing an action in that muscle. If we don't want to cause an action, we just stop sending signals. Now, there's two types of synapses. Remember, a synapse is just the connection between the neuron and whatever it's connecting to. So whether it's another neuron or another muscle or a target organ. So you could have an electrical synapse or a chemical synapse. Electrical synapse occurs when you have two axon terminals that are right up against each other, and they're so close that we have what are called gap junctions between them. And literally, the signal just jumps right across from one axon to another. Now, on the other hand, a chemical synapse uses neurotransmitters. These are chemical messengers that are housed in these vesicles in the axon terminals. And when a signal arrives here, it actually tells these vesicles to release the neurotransmitters across the synapse to bind with a receptor. So they bind with a receptor to produce whatever action it is that they're supposed to produce. So remember, there's two possible general actions, excitation or inhibition. So a few examples of excitatory neurotransmitters are norepinephrine, also called noradrenaline, dopamine, and acetylcholine. So each of these has their own specific type of receptor as well. So here's what excitation looks like at the synapse. The action potential arrives at the end foot or the axon terminal, and it causes calcium to rush into the axon terminal. And that calcium stimulates these vesicles to release the neurotransmitters across the synapse. So the neurotransmitter then arrives on the other side of the synapse and it binds to the receptor that's specific to that neurotransmitter. When that happens, it actually stimulates a new action potential to be started on the other side. Now, if this is a neuron, that is going to send that action potential down the length of the axon on its way to wherever else it needs to go. If it's a muscle, this action potential is actually going to stimulate contraction. Now, make sure that you check out the muscle contraction lesson 2 to see what happens there. Now, with inhibitory neurotransmitters, the whole process is the same right up to the point where the neurotransmitter binds to the receptor. When it binds to this receptor on the other side, it, instead of causing depolarization and a new action potential, it actually causes hyperpolarization, and therefore it stops this action potential. So it can't propagate, it can't conduct any further, it inhibits the signal from continuing. So two common inhibitory neurotransmitters that we see are serotonin and GABA. Again, this is only in neurons and not in the neuromuscular junction. Now, one quick thing to know is that there are other exogenous chemicals, and exogenous just means outside of the body, that can have an effect on this process at the neuromuscular junction. So again, between nerves and muscles. So that just means that our nerves and muscles can be doing everything right on their own, but if these chemicals are present at the synapse, it changes what happens. The first one is atropine. Atropine can actually inhibit acetylcholine. So acetylcholine gets released, it goes across the synapse, it tries to bind, but atropine comes in here and inhibits it from working. So acetylcholine gets released, but can't actually do its job. Now, the other one is neostigmine or physiostigmine. These are drugs used in a condition called myasthenia gravis that will actually inhibit acetylcholine esterase. Now, acetylcholine esterase is an enzyme, and you know it's an enzyme because it ends in ACE. So this is an enzyme that actually deactivates acetylcholine once it's no longer needed. This is a normal process. Acetylcholine gets used, acetylcholine esterase comes in and deactivates it so that we don't have any overactivity. But when neostigmine or physostigmine are present, this enzyme can't do its job. So that means we're actually going to have acetylcholine continuing to stimulate these nerves, and it's going to have this increased effect of acetylcholine. Make sure that you check out the lesson on myasthenia gravis to learn more about why that is actually a good thing. So let's review nerve transmission. Remember, it could be an electrical synapse using gap junctions, or it could be a chemical synapse using neurotransmitters. And that could cause either an excitatory signal to keep the action potential going or an inhibitory signal to actually stop the action potential altogether. And just remember, there are many, many, many specific neurotransmitters that each have their own receptors and their own specific actions at the synapse. So that's it for the A&P of nerve transmission. Make sure that you check out all of the resources and associated lessons attached to this lesson. Now go out and be your best selves today, guys. And as always, happy nursing.